Good morning, everyone, and welcome to this free hands-on webinar on NGS Omicslogic training. To start today's session, I want to invite Dr. Singh to introduce the speakers.
Okay, thank you, Bipsa. First of all, I would like to thank Pine Biotech for connecting JUIT to your wonderful platforms like Bioinfo and all that we are going to learn today, because I did this exercise especially for our students of BTech and MSc Biotechnology and BTech Bioinformatics, and some PhD scholars will also be there, and some of faculty members of our departments are also here. So before starting anything, first I would like to call upon our HOD sir, Professor Sudhir Kumar, to please brief about the department, and then we will proceed further. Professor Sayal.
Dr. Tiratha, am I audible?
Yes, sir.
Okay, thank you. Good morning, everyone. And my special thanks to the Pine Biotech team and all the members who are here, Dr. Mohit, Dr. Harpreet, and Ms. Pipsa. So thank you for arranging this webinar on NGS and Omicslogic training for JUIT, Biotechnology and Bioinformatics students. So on behalf of my department and university, we are very thankful to you for this webinar, and also a good effort by our bioinformatics faculty, Dr. Tiratha, who is actually linking up your team with the potential students. And I'm very sure that this webinar will be very helpful for the students in large and also for the faculty of the department. Just to introduce you about the department, because probably Dr. Mohit, Harpreet, and Bipsa, you might be connecting us first time. This department, which is the Department of Bioinformatics and Biotechnology, like established in the year 2002, and the first program that we started in the department was actually BTech in Bioinformatics. And I'm very happy to tell you that this is one of the oldest program in the country. Like since 2002, we are running this BTech program in this university. And then later on, we added BTech in biotechnology and then MTech in biotechnology. And also we have very good program and strength in PhD bioinformatics and PhD biotechnology. And in past, we also had, even in now, we have research projects in all the domains related to bioinformatics and biotechnology. And we are 17 strong faculty member in this department with many scholars and undergraduate and postgraduate students, and even a few new batches of MSc biotechnology, they also joined. So I'm very sure that once this pandemic is over, we would like to have you here physically present and a one-to-one meeting with our students, and that will be great. So with these words, I again welcome you and very thankful to you for your kind time and for arranging this webinar. Over to Dr. Tiratha. Thank you so much.
Thank you very much, sir. So, my dear fellow faculty members and my dear students, this webinar we have organized especially for students so they can learn, they can connect with the Pine Biotech, which is doing really good in the big data analytics and data science for biological and biomedical domains. And today we have two speakers with us. So first I'm introducing Dr. Mohit Majumdar. He did his PhD from JNU, his master's from Jamia Millia Islamia, and having more than 10 years of rich research and industry experience. He has been co-author for like more than 25 international peer-reviewed articles, 3 book chapters, and various international conferences. He has been instrumental in developing bioinformatics facility at the School of Life Sciences, JNU, and is also a recipient of multiple awards like Indo-Sastri Award from Government of India, NVIDIA GPU Award, Fujitsu Award, and many other awards. He has 5 years of teaching experience working as teaching assistant in JNU, where he was teaching MSc and PhD students of bioinformatics. Currently, he is the head of global business development for Pine Biotech, USA, and also contributing to marketing and sales. He is also mentoring participants from various programs. So this is about one of the speaker, Dr. Mohit Majumdar. Now I would like to call upon one of our BTech third year student from bioinformatics domain, Bhumi. She is going to introduce you, Dr. Harpreet Kaur. Bhumi, over to you.
Thank you, Dr. Tiratha. Good morning, everyone. I'll be telling you about Harpreet Kaur. Harpreet Kaur has done her PhD in Bioinformatics from the Bioinformatics Center, CSIR, Institute of Microbial Technology, India, and JNU, New Delhi, India. Her doctoral research is focused on the domain of cancer genomics, specifically computer-aided identification of genetic biomarkers for predicting liver cancer and its prognosis. She nearly has 7 years of research experience and 1 year of teaching experience. She received her master's degree in molecular biology and biochemistry from the Guru Nanak Dev University, Amritsar, Punjab. Her core field of research is cancer genomics and machine learning, and expertises in the application of statistical machine learning techniques on biomedical data and development of prediction tools and databases. Her current research interests are in the area of artificial intelligence, machine learning, cancer genomics, biomarker discovery, and database development on different aspects of human health. Harpreet has published 30 research articles in peer-reviewed international journals and over 200 plus citations on Google Scholar. She has received the EASL Young Investigator Bursary Award from the European Association for the Study of the Liver to present her research in the Digital International Liver Congress 2020. She has received an INSE Young Researcher or Achiever Award 2020 from the Institute of Scholars. She currently possesses a membership for various scientific sorry.
Hello, Bhumi.
Dr. Singh, is Bhumi's.
I think there is a audio loss, so maybe we can.
Yeah.
I mean, the introduction is complete, I guess.
Yeah.
And we can proceed.
Yeah. Anyway, thank you, Mohit, and thank you, Harpreet. Now platform is in your hand. Please proceed. Thank you very much.
Thank you, Dr.
Thank you.
Yeah.
Thank you, Dr. Sudhir, for the introduction. Today, we will discuss the changing landscape of data-driven research in life sciences and the opportunities that come with the widely accessible high-throughput technologies. We will learn about various data types and these emerging technologies, which have shown tremendous impact on healthcare, agriculture, environmental sciences, public health, and more. Before we start today's session, I want to introduce you to Pine Biotech. Omicslogic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and the Tauber Bioinformatics Research Center. What we will discuss today is a result of hard work by an amazing team led by company co-founders Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at the University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career, has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team you see here includes bioinformaticians, computer science experts, and we will be hearing from some of our team members today who were introduced, Dr. Mohit Mazumdar and Dr. Harpreet. I also want to introduce you to our CEO, Ilya Brodsky. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. Julia Panov is an Omicslogic mentor and an expert bioinformatician who is also responsible for developing the resources we use in our Omicslogic programs. And we have already heard about Dr. Harpreet and Dr. Mohit. So now I want to invite Dr. Mohit to take over today's session. Over to you, Dr. Mohit.
Thank you, Bipsha. And let me share my screen. Hello, everyone, and a very good morning to all. My name is Mohit Mazumdar, as you have already got the introduction, and I just wanted to start by saying that I did my master's in bioinformatics from Jamia Millia Islamia, and then I went on and did my PhD from Jawaharlal Nehru University, New Delhi. But just to share this, because it's a pleasure to have you, Dr. Sudhir, and Dr. Sudhir mentioned about the university. And I just wanted to say that my first ever conference that I attended in bioinformatics was in Jaypee Institute of Information Technology in Noida. So I felt somehow connected with Jaypee Institute. To talk about bioinformatics education and research, in my current role, I'm collaborating and working with scientists and researchers for bioinformatics research. And we are trying to collaborate with the universities in India to provide support for bioinformatics education and research and international collaborations, fellowships, student exchange programs with our partner universities in the US. So in this 12 years of experience working and collaborating, I have, before Pine Biotech, I worked for a company called Accelera, and that company, in that company, my role was to collaborate with pharma industry. And in that role, I have to collaborate with pharma industry to offer bioinformatics services and understand that what are the bioinformatics need for the industry as such, especially pharma, big pharma industries, small pharma industries, and biotech companies. So that is about me, but I would love to know, about the participants who have joined this session. So it's an interactive session that we will be doing today, and it has an hands-on aspect as well. So to do that, let me ask you a question, and if you can answer this in the chat box, it would be really great. So to know you, I would just like to ask the degree that you're doing, and if you're doing a specialization, if you're doing a master's, if you're an undergraduate, if you're doing a PhD, or if if you're doing a postdoctoral or research. So if you can just write it down in the chat, that would be really awesome. So I did my PhD. So if I have to write, I'll write PhD bioinformatics, or yeah, that's my specialization. And it would be great to know you all and know what you are doing and what you are interested in. Thank you, Akanksha, for sharing. You're doing BTech and you're in fourth year, bioinformatics. And Ishita here is doing biotechnology, and she is in second year. Malik is in third year bioinformatics, Kaushal bioinformatics. So I see there are a lot of students that are doing bioinformatics, and I know that bioinformatics have been a prominent domain, and that the university is working on, and really glad to see so many students doing bioinformatics undergraduation. Thank you. Thank you, Swami. Thank you, Sharavi. Thank you for sharing. And it's great to have you all. And it gives us an opportunity to learn about you, because this webinar that we are doing and this entire collaborative platform that we have developed is helping clinicians, biotechnologists, biologists, and bioinformaticians as well to learn about big data, NGS data, and the application of this NGS data with ease. So our purpose is to simplify bioinformatics. All right, thank you all. Thank you for sharing. Moving on. So the discovery of structure of DNA had a profound effect on the ability to study genetic code, right? However, it took almost 50 years from that time to sequence the first human genome. Soon after the technology developed, it started to evolve rapidly. So by 2015, the 1000 genome project came, and before that, there was a revolution in terms of the technology that has been developed and have evolved throughout that process. So after reaching that stage where we have a large number of genomes available, in 2008 there were 100,000 genomes sequenced. And today, there is an ever increasing number of genomic, transcriptomic, proteomic, and metagenomic datasets that are available that describes the subcellular processes with highly detailed digital data. So before learning about the omics data analysis, it's fair to ask why we want to learn about big data bioinformatics and its application in modern and translational sciences. So today's session will introduce you and include an introduction to Omicslogic resources, followed by some hands-on practice. So as we are starting the session, it would be nice to know how many of you are on laptop, desktop, cell phone, or others. Because for hands-on session, we recommend you to use either laptop or desktop. But if you are on cell phone, we are recording the session and you can practice later as well. So if you can please write it down 1, 2, 3, or 4. I mean, we would know that yeah, Rohit, thank you. So Rohit, you're on laptop. Ishita is also on laptop. So a lot of people on laptop. That's great. Even if you're in mobile, if you're using mobile, we will be recording this session, and we will be able to access the resources that we discussed today. And thank you for sharing. All right. So as I was telling you, that it's fair to say that, or ask why we want to learn big data bioinformatics. So biology as data science. Well, what we are seeing is a transformation, right? I don't think that it would be surprising if I say that biology is rapidly acquiring the characteristics of data science. Billions of data points on genes, proteins, and other molecules are compiled in in large files and libraries and databases, and are being systematically studied. This influx of data, associated methods, and data-driven research makes bioinformatics for life sciences as prominent as an emergence of computers for the tech industry. So analyzing this data will lead to more knowledge and understanding about living organisms. This introductory course or session is designed for an overview of bioinformatics for students, biologists, and clinicians. So big omics data is shaping biomedical innovation. where molecular data is used for precision diagnostics, data-driven integration, and machine learning is transforming personalization of treatment selection. Innovative algorithms allow us to do this unprecedented capacity or capability to do drug discovery, design of large molecules, and drug repurposing. So in research, if you see, when you're doing research, and I was involved in a lot of bioinformatics research throughout my career, so I know that for a fact that we do a lot of mechanistic studies when we are learning about a single protein or mechanism of a disease. And when we are doing that, we are doing a lot of bioinformatics analysis by modeling, by looking at the available data, and then generating information or predicting based on the results that we have seen from our wet lab analysis or the literature that has been published. So bioinformatics is heavily used, and in today's world, most of the publications that you will see have a component of bioinformatics. In clinical settings, bioinformatics is playing a bigger role. And I'm saying that because of my association with pharma and the way I have seen bioinformatics is being utilized by companies. So patient stratification is one of the critical or key area for personalized medicine. So we are dividing patients into groups based on their genetic information, disease variations and comorbidities, identification of therapy responders, mechanism of disease and disease progression. These are all studied using the data. In pharma and biotechnology industry, where it is driven by innovation, biomarker discovery plays a huge role. Detection of toxicology, molecular mechanism of drug action based on the literature and based on the information that we get from network analysis and all those things. Drug target discovery is something that we have heard so many times that this is an application. And drug repurposing and repositioning is upcoming field, because we have now a list of FDA approved drugs that have shown quite a good effect on other diseases as well. There are some prominent examples of that as well. And when we are discussing about current pandemic and the application of data driven research and the outcome that we are seeing, vaccine research is a big field in that. So if you see the jobs that are posted most frequently, research that is ongoing, data that has been generated by the clinicians, the current nature of application demands you to understand some of the critical steps that are changing the world of healthcare, including the understanding of biological data and how to interpret and what to expect from it. So there are some there are some biggest companies on pharmaceuticals and healthcare that are now using data-driven research, and these companies now have huge teams who are working on data itself. And I know for a fact that the company Novartis, I have few friends there, and they have transformed into a data-driven company using multiple information and genetic information and clinical information that they have collected from, hundreds of years. I know for a fact that people who have submitted the information way back in 1950s, and those information is now converted into a digital format so that data scientists can use that information. One of the other example is Regeneron, and I think you must have heard about this company. For example, just this is a page from their website, and you can see that how much genetic information they are collecting, and they are using this information that impacts human health and plays a huge role in novel drug discovery because of the complexity that we have understood after the Human Genome Project, of course. And when I talk about the current pandemic and the COVID vaccine, and as Regeneron is one of the company that is working, they are using a lot of data, patient data from these studies to help come to these conclusive or discoveries. So it's what the purpose of these slides are, to let you know that there's a lot of opportunities that are related to data science in biology, or biology as data science. And the biology is now transforming, the modern biology is now transforming towards a data driven research. So it's clear that the rapid growth of high throughput data has given rise to data driven discovery in life sciences. And as a result, there's a significant demand for data driven skills, data science skills, and experience with bioinformatics method of analysis. But what are the challenges? Because we talked about the importance of this, but there are several challenges to learn this and to learn this well. So every day, sorry, every day we are working with students, researchers, and faculty around the world, around the globe. And what we are doing, we are simplifying bioinformatics and overcoming the difficulties with innovative curriculums, cloud-based infrastructure, to overcome the burden of having to overcome the burden of getting these sort of computational infrastructure. Because when we are talking in about big data, we are talking about data in megabytes, in gigabytes, in terabytes. So when you, if you want to analyze that sort of data, you would require a computational infrastructure. And to make this available to everyone, one of the challenges is to bring it to a medium with which everyone can access these resources. So a cloud-based platform is the answer. And we've been also developing a content in terms of supplementary study materials, courses, and research-based projects that you will be seeing in this presentation. So let's first review some of these resources. So one of the resource that we recommend everyone to take is a free resource, which is Introduction to Bioinformatics. Introduction to Bioinformatics is a introductory course that is available on our platform and which is free for everyone to take. This way of learning is called asynchronous. These are asynchronous courses, and the way of learning is that you are learning at your own pace. So the coursework that have been provided in this introductory material have been taken by thousands of people around the globe and have been validated. And these online courses cover important terminologies and provide perspectives from experts on this field, on this exciting field. And each course you will see on the platform, and this is a free course that I'll encourage everyone to take, has quizzes that allows you to see how much you have learned. So all of this activity is measured using a point system that lets us identify any technical issues and resolve roadblocks that are so common for bioinformatics, and a certification from our program mentor and Tauber Bioinformatics Research Center and Pine Biotech and the university for all the hard work that you're putting in. So if you ask what is project-based learning? So project-based learning and project-based learning and research-based learning is our key focus. It is an experience of learning real application of bioinformatics, because what we do, we take data sets from high impact publication and use them for practical exercises and developing research projects. So what we are doing is that we are going through the literature on the publications that are coming up on different uh, biomedical area of research, like COVID-19, like malaria. We are working in Africa with African universities, and we are trying to help them learning about Ebola virus and the outbreak with Ebola virus. In India, we are working with several institutes on tuberculosis. So one of the way of learning this is to learn via data and publication, which has already been, it's a well digested research in the scientific community. So focus and specialization. the application of bioinformatics, and I am saying this a lot because while doing my PhD, while doing my master's in bioinformatics, I realized that I've been working on several problems, but the application of those problems are not so much translational. So I was always inquisitive about doing something that has a real effect on the society, or if it has a real effect on the healthcare system. So one of the areas that we work here in Pine Biotech is uh, these translational areas. For example, infectious diseases, neuroscience, oncology, precision oncology, and as I was telling you, the application of bioinformatics in pharma and in research. So these are I've been able to do this because of the big data, the NGS data. And the NGS data, it's so descriptive and it's so complex, but yet it gives us an amazing insight into the problems that we are looking at. That's why it's fascinating and exciting. And I think all of you would agree to that once you will go through this entire, exercise and learn about this technology and its application. So as a result of this training that we have been doing, anyone starting on their journey in bioinformatics can start developing a project of their own. So this is a big step. Learning about something and actually transforming that knowledge into a project is one of the critical and key steps. So when you go for an interview, when you go for a job, one of the things that have been discussed in those interviews and jobs are the projects, because projects are the real application of your learning. So our focus here is project based training, so that the real application of bioinformatics is something that you get experience with. So these all these resources and all this the introductory course is something of a starter a starter thing for you to start with, and then to learn about the application of bioinformatics in various fields. Like I discussed pharma, I discussed healthcare. In defense, there is application of bioinformatics. So in today's world, the application of bioinformatics is pretty big, and many of us have realized it and is doing and many of the participants are working with us on a regular basis. Many of our universities are working with us, and we are working collaboratively in this bioinformatics community to bring awareness about the, the the data that is out there and the application of that data. So today, as I said earlier, this session will include practicals. The first platform that we are going to explore is the educational platform, and the educational platform have these examples to learn from. So one of the things in education or in research or in while doing teaching, we found more effective is to learn from examples. So a publication is an example. So what we did is converted a publication into a tutorial so that it's easy to learn, because sometimes and most of the time what happens is that we are going through publication and it's really difficult to comprehend that publication, because there are so many different authors that have written those papers, and they have their own way of expressing. So sometimes it's really easy to go through a publication, and sometimes it's really difficult to go through a publication. It's also because these are complex analysis. When we are talking about a genome, that means the entire genome. So 3.2 billion base pairs in terms of if we are talking about the human genome. If we are talking about the transcriptome and the number of genes that are there in the transcriptome, that's 20,000 and 22,000 genes. So it's a quite a big amount of data to comprehend from, and that's why these are helpful, because if we learn from examples. So the first step, as I was telling you, is to access some of the free resources that are available and that we encourage people to go through to get aware about the technologies that are available. So to take you to this free to this platform and get you going with this registration, I will pass it on to Deepsha. She will be able to help you get registered and start this analysis today, the hands-on analysis that we'll be using in this session. So I'll pass it on to Beepsha now. Beepsha, if you can take our participants to the educational platform and show them around.
Yes, sure, Dr. Mohit. Let me just share my screen. Okay, give me a minute. I hope everyone can see my screen. So I have also pasted this link in the chat. I will be pasting it one more time for everyone. Please come with me to the educational portal. So once you are here, you will notice that there is a register or login button for you. If you're not already registered, because I'm already registered, it's showing my profile. So I'll just also show that. So you just have to click on the register button if you are registering for the first time, and you will have several options from which to register for this site. And this is a free registration, and once you're registered, it is a registration for a lifetime. You will be able to access all of the free resources that we make available on this portal. So just to give you all an overview, this portal will have a lot of information on the different things that we offer. So now starting right at the top, you see here are the courses. So Dr. Mazumdar has just told you about Introduction to Bioinformatics. So let us look at Introduction to Bioinformatics here. And also let me know if you're able to register and log in the chat box. If you're already registered and logged in, please put number 1 in the chat box so that I come to know that at least a few of you are here. I will be waiting a minute or two for everyone to come and start, especially those who mentioned that you already have a laptop with you. If you're registered, please put 1 in the chat. Okay, Ishita, great. Thank you, Jigyasa. Maybe a few more people so we can do this together. Thank you, Ramnik, Ria, and Aditi. Okay. So you can follow along. So as I showed, if you go under courses, you'll find Introduction to Bioinformatics. So if you click on Introduction to Bioinformatics, you will be given the option of take this course. So as you see here, this course comes with different sections. So here, first we have an introductory video, which will introduce into what Introduction to Bioinformatics is. Next, it shows me retake the course because I have already taken the course, but for you, it will be starting. So in the overview, you will have an overview of the course description and the course features. So you will see how long it should take to finish the course, how many lectures are there, and whether there is a certificate or not. And this too, I will be showing you how you can access your own certificate. So once you come to the overview, there will be the curriculum. So in the curriculum, you will find that to start with it, you have an introductory survey. So what this introductory survey is, here you share information about yourself with us. So for example, maybe you are interested in cancer research, you may be interested in neuroscience. So you can let us know about this different introductory survey which you can fill up. And we will be able to learn more from you and more about you and then help you out with this. And once you finish the introductory survey, you will be coming to Introduction to Bioinformatics. So once you come to Introduction to Bioinformatics, you will see that the lesson starts. So please go ahead and fill up the introductory survey here. So you see, this is a video And there is also text and images. So the way these courses are designed for self-study is that you will be coming across videos, text, as well as these images to help you learn using all of the different senses that you can employ. And also online sessions like this are here to help you get guidance from mentors who will be providing you the right path to take for the different kinds of research that you are interested in doing. So if you go on down, once you finish each lesson, then you can go to the next one. So this way you can complete this entire course. And like Introduction to Bioinformatics, there are several other courses as well on our platform, and the entire course collection can be viewed here. And these are actually divided on the basis of topics, which is, or multi-omics, which is the kind of research areas that we teach. So you will have Introduction to Bioinformatics here, then Epigenetics. Now, each of these sections will have different number of courses. You have machine learning in BioML, metagenomics, genomics, transcriptomics. So let me just give you an example from transcriptomics. Like transcriptomics has 4 courses within it. So it's not one single course, but 4 courses broken down that will teach you about RNA-seq analysis. So you can go and explore these courses as well, but not all of them are free. So I will come to these later on. Okay, so this is how you can keep on exploring the platform. We also have a blog which we offer where we share a lot of our free resources. So you can also go and explore the blog section. I will also put it down in the chat box for everyone for easy access. So the blog will also have a lot of topics on different areas, and we keep on sharing a lot of resources. So the blog is always open and free. You can explore the blog. And before I pass it back to Dr. Mohit, I just wanted to show you how your profile looks like. So for everyone who has registered, you need to go ahead and update your profile. Like my profile has a profile photo, it has my full name. You can add your social media links as well. Especially, we would recommend you to add your LinkedIn IDs, because LinkedIn is a place where you can connect with a lot of your future employers, prospects, and collaborators. So that is a very good place to start with. So each of the courses you do, like if you finish Introduction to Bioinformatics, you will be earning points. So you see under my profile, the points are mentioned here. So I have over 30,000 points. And how to update your profile, if you go under your own profile, you will find there's an edit button with which you can use to edit your profile and update it. So I would recommend that even if you're not doing it right now, you can go ahead and create your own profile and get started with the different courses. Now, once you have done the different courses, it will show up under your profile. It will show up under here. So this is your own profile section. You will be able to see all of the different activities that you are doing. So once you come up here, you see the course that I completed will appear as completed. The course that I'm taking will appear as the percentage of the course that I have only completed right now. So once I finish with a course, my certificates will also come up here. So these are all individual course certificates. So if you are doing more and more courses, you will be earning more and more certificates. So having said this, you see here are 2 of my certificates. So Introduction to Metagenomics is also a free course that you can. So now I would want to pass it back to Dr. Mazumdar. Dr. Mazumdar, over to you.
Thank you, Vipsha, and to everyone. have a profile, and we'll be able to interact with you with your bioinformatics problems or your data-driven questions using the same profile. You can always comment there, ask us if you need any sort of guidance for your undergraduate program, for your master's program, for a PhD, or for job opportunities. Now, going back to the presentation. So the resources that Deeksha discussed today, I'm just going to elaborate on those, the specialization tracks especially. So if you're interested in learning about DNA, if you're interested in learning about the changes that can happen to the genetic code, the genomic series of courses explain how next generation sequencing can be used to study genome-wide genetic variations that results in phenotypic changes. If you want to learn about transcriptomics data analysis, here is a complete guide to RNA-seq data sequence analysis. Transcriptomics 1 is a introduction to NGS for study of RNA expression, RNA gene expression. Transcriptomics 2 talks about the conditions, different conditions, and for example, we start using some of the statistics to understand the data, the statistics behind when you get the data and you do the descriptive statistics to learn about the preliminary data, and then you start exploring the data. Transcriptomics is very interesting because it's dedicated to advanced analysis methods of transcriptomics data by using machine learning algorithms that allows us to find meaningful patterns in big data sets, especially complex patterns that are typical for RNA-seq experiments. Transcriptomics 4 is a review of single-cell RNA-seq. Single-cell RNA-seq has been demonstrated as a really powerful technique for the classification of tissue-specific cell and the study of time course data for thousands of single-cell samples. So you can learn about that in Transcriptomics 4. And all of these courses have hands-on application. So you'll not just be learning theory. So you'll also be doing the experiments yourself in using these courses. Metagenomics. If you're interested in metagenomics and if you are interested to learn and dive into the world of microbiome, metagenomics will familiarize you with the way microbiome influences their host and compose a unique community. So that we're going to talk about the unique bacterial, viral communities that we can study using next generation sequencing, and the impact of bacterial diversity, microbiome on human health, and learn about different sequencing methods like 16S ribosomal sequencing, RNA and DNA, learn to compare microbial composition in different conditions. So the application of metagenomics is huge, and I think we also have an upcoming program on metagenomics, which we'll be talking about later. Machine learning. So many methods in bioinformatics would require an in-depth understanding of various machine learning algorithms and tools. So in this specialization course on machine learning, we developed a series of courses and practical tutorials that provide a solid foundation for students on these important and often misunderstood approaches. In this series, student will learn about computational innovation and algorithmic improvements that are the forefronts of this biomedical discovery. And as I was telling you about the project examples, so by seeing how experts have analyzed their data and achieved research results is an important aspect of this entire research journey or the learning that you're getting. So identifying the appropriate type of data and planning your analysis. So all of them, this entire platform, the entire educational platform is built to help students, is built to help faculty, so that faculty can take these courses and teach it to students, take these examples from different publications that are made into tutorials for easily learning this complex, research. As you see the titles and the application. It is in agriculture, it is in virology, it is in neuroscience, it is in cancer, it is in biotechnology. So the application is everywhere, and it's important to learn this through projects. And I think it's the best way to learn this through projects. And how do we do this, and what are we going to do today is something that we'll be covering in the next part of the session. I think we have bored you a lot with all the theory, but we'll be doing a lot of practicals now. But before doing that, I think I want to talk to you about the second platform. This is our second innovative platform that is developed at the Tauber Bioinformatics Research Center, University of Haifa, Israel. And there are several sections here which have been developed by collaborating with universities in the US. So there was a big grant that helped us to develop the virology section in collaboration with UCSF and Harvard. And there's several sections in this platform that describes different types of data. So if you see this NGS data, so we have a section for analyzing NGS data, mass spectroscopy data, structural data, and then we are integrating data for modeling. We are doing the platform has also capability to do mathematical modeling and crunch through these different multi-omics data, and then integrate these multi-omics data in a single experiment, or to get a real holistic picture of the analysis. And we are using mostly innovative machine learning algorithms that are being used to understand patterns from big data, and that is applicable for almost all the industry in today's world, from Amazon to from shopping to Facebook. Everywhere big data is being used. Okay. And then you have an interface to see what you are doing. So this is a cloud-based platform. So you do your experiments here, and you see the results of your experiments here as well. And then you will have a specific login when if you're a part of a program and stuff like that. But today we are going to do some hands-on analysis using this T-Bioinfo platform, and I am pretty sure you all be excited to do this. But before I talk about more about this analysis, I think if you have any questions, please go ahead and ask your questions. And meanwhile, Deepsha, provide if you can provide the students with the form to get the demo account to do the analysis. So if you want to follow with us and do the analysis, you're welcome to do that. Please email to Bipsha, or send us a message that you would need the demo access, so that we can do this together. And if you're doing it on mobile, then maybe we can do this later as well. So the first step of doing a hands-on analysis on a data set is to understand the data set, right? What are we doing and why are we doing this? So let's talk about a case study, and then we will do the hands-on on that specific data set to learn that what information, like today we talked about the application, what information that we can get from doing a hands-on analysis on a biomedical data or a transcriptomics data. So let's look at this example data set. Here is an example, and this publication is interesting because the authors have assembled a data set of about 50 different breast cancer cell lines that represents the whole spectrum of breast cancer variation. So the cell lines have genomic, transcriptomic, proteomic data, as well as efficacy measure of commonly used and experimental cancer drugs. So you can find it using this link, and let me share this link with you on the chat also. So this is a publication that you can look into and learn from. And as I was telling you that this kind of education, and we have been doing this for students coming from high school. So we have students from high school who have been able to do this. We have been students from undergraduation, first year, second year, third year. So it's not about like the complexity, it's about the eagerness to learn this. And it's with the platforms, it's really something that anyone can learn from. So as I was telling you this about this data, so here the data from cell lines, and it's combined with two types of phenotypes. So do you guys know what is a phenotype? Just say yes or no. Phenotype and genotype, some genetic information, and phenotype is what we can observe, what we can see, and what we can record on that basis. I think I'm getting some answers. Yes, yes, yes. That's so good. Thank you. So, as I was telling you that this pan cancer, so pan cancer data set and growth inhibition factor that has been recorded, GI50, we call it GI50, of these cells in response to different treatments, typical such as chemotherapy, targeted therapy, and other cancer drugs. And then they used machine learning, so 84 cell lines, and then molecular profiling, and then this data they have 70 cell lines, data for 70 cell lines, and the effect of 138 drugs on this cell lines. Okay, and then they use machine learning methods such as random forest and support vector machine, and then they were able to classify these patients based on the treatment response, like which drug should be given to which kind of patients with which kind of molecular profile. Is it clear? Is this clear what I've explained? If not, then let me take you much deeper about this how this is and why this is, and what is breast cancer, what are the subtypes. So let's do this little bit in more depth. So classifying breast cancers into different categories. So if I'm saying breast cancer, it means that you're thinking that it's a single cancer, but actually it's not like that. So breast cancer have several subtypes, like different types of breast cancers, right? So subtypes of breast cancer, and what are those subtypes of breast cancer, and how can you understand or predict which type of breast cancer it is? Because when you go to a doctor, when you go to a clinician, when you go to a doctor for a checkup, the first thing the doctor maybe the information that doctor wants to ask or wants to learn from you is maybe about your age. He would want to know about whether you're a male or a female, or whether what is your weight and stuff like that, because that influences the drug response, right? So sometimes a drug works differently for a male, for a female. For kids there are different dosages, for adults there are different dosages. So just think in that context that people the doctor needs that information to prescribe your drug. So the first important thing for our doctor is to do correct diagnosis. So diagnosis is very important. So unless you understand what kind of problem there is, the solution would be really difficult to find. So to understand the problem, to understand what kinds of breast cancers are out there, let's understand let's look at this example and understand. So this is a mammography. A mammogram is a X-ray picture of a breast that doctors take a look at, look for early signs of breast cancer. So in this example, you can see a tumor that is highlighted, and once it is found, it is critical to establish whether it is a malignant or benign and what tissue it is originating from. Once the tumor is characterized by its phenotype like size, tissue, origin, etc., additional information can be obtained by looking at the presence of specific proteins that characteristics of that cancer. So based on the literature study that we have done so far, we can relate, like these are the known biomarkers. If this protein is highly expressed or there, this is this is a kind of breast cancer. So many of the popular proteins that you have might have heard about is EGFR, BRCA1, HER2, Herceptin 2 receptors. So if I just go back here, you can see that if the this tumor is within this tissue, it's called ductal carcinoma in situ because it's in a ductal shape. This is in a lobular shape, so it's a lobular carcinoma in situ. So it's in ductal, but it's gone outside the tissue as well. So it's an invasive ductal carcinoma, and similarly invasive lobular carcinoma. So what is happening is that, of course, this is more dangerous because it has a probability to spread around. So about the about this entire technology or the process. So this is called immunohistochemistry or antibody staining that can be used to find if they are expressed, if these proteins are expressed, or like specific biomarkers that are characteristics of the cellular event, such as proliferation or cell death. These are typically referred to such biomarkers based on those analysis. So another aspect of learning about this molecular subtypes is by looking at the gene expression. So this is a microarray data. So we are evolving from seeing a picture to more information here and then a lot more information here, because you are here seeing 5 proteins here. Sorry. And 5 here, 4 and 5 here. So here we have a panel of 50 proteins, and with this panel what we are trying to see is the expression of genes. So there is a different pattern here, there's a different pattern here, there's a different pattern here, there's a different pattern here, and there is a different pattern here. So in this 50 genes there are many biomarkers, and there are also housekeeping genes. So housekeeping genes are those that will have a normal expression, right? So based on that, you can classify them into 5 groups. But one of the challenges in doing this is, again, like when we are learning about a person's transcriptome, we are not seeing 50 proteins or 50 expression of 50 proteins or genes. What we are looking at is expression of 20,000 genes from single patient, 20 to 22,000 genes. And if there is a new subtype, if there is a new cancer, then we will not know about it because we just know about these. So nowadays what we are doing is that we are using big data. How to deal with this data? So this is an example of a gene expression table. So a gene expression table is what you get from transcriptomics data analysis. So once you do transcriptomics data analysis, after doing the analysis and after processing the raw data, you will get a table of expression like this. And this, in this side, you can see these are number of genes with their ensemble ID. And this is the patient sample. So this could be patient 1, patient 2, patient 3, patient 4. And in patient 1, we are looking at the expression of this gene, this gene. This is the patient 1 expression of genes. This is patient 2. And here for classification, because we already know what kind of cancer these are, so we have already written normal like, basal, luminal. So these are, so you can see that here from this table we have 2 normal like, we have 3 basal, we have 4 luminal, we have 1 Claudin low, and we have 3 more luminal. So what we are trying to do, what we have to do, is to find out patterns from this data, and then we are going to use machine learning. So it's really exciting to learn about patterns, but this is a very small table. Actually, the size of this table is so big that it contains 20,000 to 60,000 genes, because in one of the data sets we used mouse and human concatenated genes. So there's a lot of genes there as well. And then there are number of samples, tens of hundreds of samples. There's not one patient, there are multiple patients with so many genes. Just imagine here you have 20,000 rows, 60,000 rows, and here you have tens of thousands of samples. It would be impossible even to load in Excel. And they have complex relationship. And the complex relationship in statistics, we say that these sort of correlation is multidimensional correlation, and we are going to talk about how to deal with this data. I mean, we get this sort of data. These kind of data are available on NCBI to do your own research. So many of the publications have addressed maybe one thing that they wanted to look at. But this data has so much information. As you can see, there are several experiments or new research that you can come up with using this data. But all you need is these sort of learning to be able to see the complex relationship of within this data. So this is a PCA graph, principal component analysis. And to do this entire process, we will do a short tutorial today, which we usually do in our Omicslogic sessions and university collaborative programs. But today what we are going to do is we are going to try to tell you that how you can interpret this kind of data, how you can use machine learning to see the patterns. And to do that, I would like to invite Dr. Harpreet to take you through the platform to help you with this analysis and show you around. So over to you, Dr. Harpreet. And if you guys have any questions regarding whatever we have covered so far, so please write it down in the chat, and I'll be available in the chat to help you with your answers. Over to you, Dr. Harpreet.
Yeah. Thank you, Dr. Mohit. So let me just share my screen. So before moving into today's session, just let me give you a brief introduction about me. So my name is Harpreet Kaur, and I have done my PhD in bioinformatics. My research was on biomarker discovery for diagnosis and prognosis of liver cancer. And in my current role, I'm Omicslogic bioinformatics trainer with Biotech, and I will be happy to assist participants to learn about data analysis, machine learning, and visualization in the application of the large data sets used in the biomedical research. So today I will demonstrate to you how we can generate a well-structured gene expression data table that already Dr. Mohit has shown you from the raw sequencing reads in a very simplest manner using the Bioinfo platform. So if you are not familiar with the coding and the programming, then you can explore Bioinfo platform. Besides, I will also demonstrate to you how you can learn coding to perform any analysis if you are interested to learn the coding and the programming skills on a Omicslogic platform. So now let me take you to the Bioinfo platform and let you demonstrate how we can generate a gene expression table from the raw sequencing read data. Just give me a second. So this is the Bioinfo platform. So my screen is visible to everyone. So this is our Bioinfo platform, where on this interface, you will find various modules like NGS data, mass spectroscopy, structural biology, data integration modeling. So just write the answers in the chat box so that I understand that you are on this platform. Just write 1 or yes if you have the access to this Bioinfo platform. So just type 1 or yes if you are there on this Bioinfo platform. Okay, Akanksha, Ria, that's great. So on this Bioinfo platform, we have a various sections for depending upon our data. So today I'm going to demonstrate how we can produce a gene expression table from NGS data. So before heading into the demo, let me just show you what are these raw sequencing data? So these raw sequencing data is present in the files that is called FASTQ files. So if you look at even a single sample, so if you look at this, here we have 15 GB data, even for a single sample. So it is impossible to load this data on your system. So how we can process this data? So let's come to this T-Bioinfo platform. So here under the NGS data, you will get an option that is transcriptomics. So under the transcriptomics, the first sub option that is RNA-seq or the parallel analysis of NGS data or microarray data. So here we will get an drop down. If you click on drop down, so here you will get 3 demo pipelines. So the first one is demo for the RNA-seq, the cell line project. Second, RNA-seq for Angelman projects. Third is PDX project. So here, as this is just a demo, so I am just going with the demonstration for the RNA-seq, which contain the cell line project data that already Dr. Mohit has explained you. So if you click on this option, the first option, so we will direct it to this pipeline. So just let me know how many of you able to find this pipeline. Just type yes if you're able to find the first pipeline that is under transcriptomics section. So just let me show you again. So if you are not getting, just go to this area of analysis. Okay, so here we will get the under the transcriptomics, we have an option that is RNA-seq. So if we click on this drop down, we will get the demo pipelines. So I'm clicking on the demo pipelines, and now I'm moving ahead. So once I will click on the start, with clicking the start, as this is a demo pipeline, so we have already uploaded our raw sequencing read data. So once I click on OK, I'm able to upload my raw sequencing read files. Those are the FASTQ files. So then the next option that is getting highlighted here, that is under the pre-processing. So here we have multiple options. So the first option that is getting highlighted
Dr. Harpreet, you are not audible to us.